The HIV virus targets T helper cells and will fuse with one by bonding its glycoproteins with the cell's CD4 receptors. After fusion, the virus's nucleocapsid enters the cell, breaks down, and releases the virus's genetic material. HIV does not have DNA inside the nucleocapsid. Instead, it has two strands of RNA, an enzyme called integrase, and two enzymes called reverse transcriptase. The RNA is converted into DNA by a process called reverse transcription, which is done by the reverse transcriptase enzyme. Once the viral DNA is formed, the integrase enzyme will carry it into the cell's nucleus and integrate it into the cell's DNA. The host is now HIV positive. The replication process for HIV begins with latency. For some individuals, latency can last up to 20 years, but the average time span is 10 years. During latency, the host can be completely asymptomatic and will never be aware that they have HIV, but the virus can still be transmitted. Eventually, something will trigger the viral DNA to end latency. The mRNAs decode the viral DNA, and ribosomes synthesize new viral proteins. The proteins assemble and form virions, which exit the cell to search for a new host cell. The T cell, one of the body's most important white blood cells, has now become a factory for HIV replication. The virus's glycoproteins mutate constantly, making it all but impossible for the immune system to ever recognize HIV and create memory T or B cells. During infection, HIV replication can create almost 10 billion virions.